Hello students, this is Ankita. Welcome to Global Online Neet. So in today's session we are going to see about eukaryotic cells. Yes, what do you mean by eukaryotic students? What is the meaning of eukaryotic? Can means true. Yes, you means true. And carry on. Carry on means nucleus. Yes. So, eukaryotic cells means the cells which have true nucleus in them is called as eukaryotic cells. Yes. So, what are the characteristics of the eukaryotic cell? It has well developed membrane bound nucleus and organized organelles. Yes, has organized nucleus and the nuclear membrane. What they include? It include protist. Unicellular eukaryotes are protist, plants and animals. Okay, includes protist, protist, that is unicellular eukaryotes are protist. Yes. So they also, what are the characteristics? First of all, eukaryotic cells are the cells which eu means true and karyon means nucleus. So the cells which have true nucleus in them are called as eukaryotic cells. Yes, what they include? They include protist. Unicellular eukaryotes are protist and they have well organized nucleus and the nuclear membrane, and they also have complex locomotory and psychoskeletal structures. Yes, have complex locomotory and psychoskeletal structures. The genetic material is organized into the chromosomes. Yes, how is the genetic material organized? In the form of chromosomes. So these are the characteristics of the eukaryotic cell. Now we will see the difference between the animal cell and the plant cell. Yes, animal cell and the plant cell are the examples of the eukaryotic cell, right? Animal cell and plant cell. Yes, we will see the difference. Is cell wall present in the animal cell? No. Cell wall is absent in the animal cell. So write the first point here. Cell wall absent. Next, cell wall present. Next is, what is absent in the animal cell? Plastid. Plastid are absent. What are plastid, students? Plastids are the color containing pigments. Yes, plastids absent and plastid present. Next comes the vacuole. Is vacuole present in the animal cell? Yes, vacuole is present, but it is very small or sometimes absent only. Most of the time is absent, but if present, it is very small. Vacuole is small or absent. Yes. Next is vacuole is central. Vacuole is central and big. central यानी plant cells a maximum coverage 90% of the plant cell is covered by the vacuole. It is so big. Now an animal cell reserve food कुठल्या पद्धतीने स्टोर केला. reserve food is in the presence of glycogen, whereas in the plant reserve food is present in the form of glycogen, and in plant cell it is present as starch in the form of starch. The form of starch. Yes. In animal cell, centriole is present, and centriole is absent in the plant cell. Absent. Okay. So is it clear, students, what is the difference between the animal cell and the plant cell? The animal cell has cell wall absent, whereas in plant cell wall is present. Yes, plastid is absent in animal cell because plastid is a color containing pigment which is absent, and in plant cell it is present. Yes, which is the plastid present in the plant cell? It is chloroplast. Yes, because of chloroplast it has a pigment chlorophyll that gives a green color to the plant. Next is the vacuole. Vacuole is most of the time absent in the animal cell, but if present, it is present in small size. Vacuole in the plant cell is central and big. It is located at the central position, and maximum 90% of the plant cell is covered with the vacuole. In animal cell, the reserve food is in the form of glycogen, whereas in the plant cell, the reserve food is stored in the form of starch. Yes, centriole is present in animal cell, whereas centriole is absent in the plant cell. Is it clear? Yes, students, the difference between. So now we are going to see about the cell organelles in the eukaryotic cells. Yes, what are the cell organelles in the eukaryotic cell? First of all, plant cell has been divided into cell wall and protoplast. Yes, now let's see the structure. Before going to the cell organelles, we will see that plant cell is cell wall and the protoplast. Okay, what is protoplast, student? If this is a cell, the inside everything mixture is nothing but the protoplast. Yes, this protoplast is again divisible into plasma membrane and protoplasm. Okay, now this protoplasm is again divisible into cytoplasm organelles. These organelles are again of two types. Yes, membrane bound and non-membranous, that is non-membrane bound. Is it clear, students? See what is happening. Plant cell is divided into two, that is cell wall and the protoplasm. What is the protoplasm? The inside cell mixture, whatever it is nothing but the protoplasm. This protoplasm is divisible into two, that is plasma membrane and protoplasm. Then this protoplasm is again divisible into the cytoplasm and the organelles. Now these organelles are of two types, that is membrane bound organelles and non-membrane bound organelles. So what is the example of non-membrane bound organelles? It is ribosome and the centrioles. Please write it down simultaneously. Ribosomes and centriole. Is it clear? Now these are the cell organelles in the eukaryotic cells. What are they? Cell membrane, cell wall, endomembrane system. What 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 all is included in the endomembrane system? ER, that is endoplasmic reticulum, Golgi apparatus, lysosomes and vacuole. You can also write this endomembrane system as GERL. That is, G stands for Golgi, ER is endoplasmic reticulum and L is lysosome, because vacuoles are most of the time absent in the animal cell. Next comes the mitochondria, then the plastids, ribosome, cytoskeleton, cilia and flagella, centrosome and centrioles, nucleus, microbodies. So these are the cell organelles present in the eukaryotic cell. Next is the cell membrane. Yes, what is the cell membrane, student? It is a universal membrane present in plant cell fungi. Yes, this cell membrane is living. How is this cell membrane, student? It is living. Just write the characteristics of the cell membrane. Fungi, it is living, it's dynamic. Yes, the appearance of this membrane is not smooth. It is a selectively permeable membrane and helps in cell to cell interaction. Please write down the characteristic of this membrane. Universal membrane. Yes. Next is it's living. How is the fluidity of this membrane? It is semi or quasi fluid. Fluidity is semi or quasi fluid. The appearance of this membrane is not smooth. It is asymmetric. How it is? Asymmetric. Next is it has patchy appearance. Okay, selectively permeable. See what happened, chemical studies on the human RBC show that the cell is composed of a lipid bilayer protein and carbohydrate. Yes, the cell is composed of the lipid bilayer protein and carbohydrate. Which lipids were included in this? Mainly phosphoglycerides were included in the lipids, which have outer polar head and inner hydrophilic tails. So the non-polar tail of this hydrocarbon is saturated and it is protected from the environment. Okay. Now coming to the cell membrane, they have studied this on the basis of the fluid mosaic model. Yes, in this, if we see that ratio of protein and lipid varies in different cells. In human RBC, the maximum, what, in human RBC के membrane में students, this question, okay, please write it down, make it a star question, कि in human RBC, in RBC of human के membrane में, how much is the protein quantity and lipid quantity? Lipid is, if this is protein and this is lipid, the protein has 52%. How much is the protein? It's 52%, whereas lipid is 40%. Yes, and rest all is the cholesterol and the oligosaccharides. Is it clear? It comes as a direct question, students, that in the RBC membrane, that is in human RBC membrane, the protein content is 52%, whereas lipid is 40%, and rest is the cholesterol and the oligosaccharides. Now, based on this ease of attraction, membrane proteins are of two types. Okay, they are called as two integral proteins and the peripheral proteins. The integral proteins are the proteins which are partially or totally buried in the membrane. The word integral itself tells us that it is inside, partially, आधा, सेमी, एक तर पूर्ण पण आत नाही आणि एकदम बाहेर पण नाहीये. Okay, they are partially or totally buried in the membrane, but and whereas peripheral proteins are the proteins that lie on the surface of the membrane. Okay, you can see in this diagram, right, हे fluid mosaic models diagram दिलेला आहे. See, look at this. This is the first phospholipid. Just a second. You can see in this diagram, students, this is the phospholipid. Yes, this is the protein. This is also protein, right? These proteins are in integral proteins because they are totally buried in the membrane. Yes, and this protein is the peripheral protein because it is lying on the surface. Yes, now as I told you, the rest is covered by the cholesterol. The membrane only has 52% of protein and 40% of lipid. Yes, this is the phospholipid. Now this phospholipid has head and the tail part. Okay, this phospholipid has phosphate head and fatty acid tail. Okay, this phosphate head is hydrophilic in nature. Hydrophilic यानी क्या students? It is water loving. Okay, how it is? It is water loving, but the tail part is the non-polar. It is hydrophobic. Yes, it is hydrophobic. That means water hating. Now, what is the role of this integral protein? Integral protein, or it is also called as transmembrane protein. It is for extraction and transport of the substratum. Yes, what is the role of this integral protein for the extraction and for the transport of the substratum? This chain is of glycoprotein. Yes, it is acting as a antigen. Yes, students, you can see this chain, green color, it is acting as a antigen. And cholesterol is here for the stability of the membrane. यानी आज तक हमने सुना है कि cholesterol is harmful. Yes, छोले भटूरे, then if we are consuming or eating any oily food, why I gave the example of chole bhature, because it's very oily. So cholesterol बढ़ रहा है, but cholesterol is also playing a role in stability of the membrane. Is it clear, students? See, this phospholipid is having two parts, right? Agar if we are drawing in simple terms, this is the head region and this is the tail region. Yes, these are, the head region is polar, yes, and the tail region is non-polar. Yes, polar region hydrophilic, okay, this polar region is hydrophilic, that means it is water loving, and non-polar region is hydrophobic, that is water hating, that means it is amphitic in nature. How it is? Amphithetic in nature. Yes. So the fluid mosaic model, this model, who proposed this model was given by Singer and Nicholson. Singer yani, don't give the name of any singer, okay, Sonu Nigam or Shreya Ghoshal, no, this model was proposed by the Singer and Nicholson in the year 1972. Yes, please remember the year. So what was according to this model? According to this model, cell membrane was made up of lipid bilayer plus protein. What was the role about this model? What they told? The cell membrane is made up of lipid. Okay, how is the lipid nature? It is bilayer plus protein. They also gave a line by Nicholas also gave a line that proteins are like iceberg in the sea of lipid. Yes, you can see in this diagram that lipids are everywhere and the proteins are in between. So what they first of all told, that cell membrane is made up of cell membrane is made up of lipid, that is bilayer plus protein. Yes, students. Next, Nicholson gave a line that proteins are like iceberg in the sea of liquid because proteins are floating in the sea of lipid. You can see in this diagram कि lipids are everywhere present and just proteins are present in between, that means they are floating. According to this, quasi-fluid nature of lipids enable proteins within the overall bilayer. How this protein can move? They can move left or right, or so called as laterally. No rotation is seen in this protein, so that's why it cannot show the flip-flop movement. Is it clear? See, काय आहे की प्रोटीनची क्वासी फ्लुइड नेचर असल्यामुळे लिपिड काय करताय, लिपिड फक्त लॅटरल मध्ये, because protein is just moving left, either left or right, or left and right होत आहे, so called as lateral movement. रोटेशन दिसतंय का या प्रोटीन मध्ये? नो रोटेशन इज सीन and it cannot show the flip-flop movement. This ability to move within the membrane is measured as its fluidity. तिथल्या तिथंच ते मूव करतायत. That is called as fluidity. Is it clear, students? Now what are the functions of the cell membrane? First of all, it transports the molecules. Yes. What is next? It is a selectively permeable membrane. म्हणजे काय सगळ्यांनाच आत येऊन देते का? नो, इट अलाउज सम ऑफ द मॉलिक्यूल्स टू गेट इनसाइड. येस, ड्यू टू द फ्लुइड नेचर प्लाज्मा मेम्ब्रेन कैन हेल्प इन आता of the intercellular junctions, secretion, endocytosis, cell division, etc. Yes, these are the functions of the plasma membrane. First of all, what is the main function? Transport. Yes. Then interaction with the cell. Then for growth, for formation of the intracellular junctions, and also the compartmentalization. You can see in this diagram, compartments are being formed. Then the endocytosis. Endocytosis म्हणजे काय? It is the eating movement of the cell. Eating movement. Yes. In this eating movement दोन टाइपचे. सेल काय करतो? दोन टाइपचे eating movements दाखवतो. One is the phagocytosis and the other is the pinocytosis. अगर फैगो है यानी क्या? सेल खाना खा रहा है. फैगो फॅगोसायटोसिस म्हणजे काय? की सेल ईटिंग. आणि पिनोसायटोसिस म्हणजे सेल ड्रिंकिंग. ओके, that means in one case cell khana kha raha hai, in one case cell is drinking water, means drinking any fluid. So these are the two movements of endocytosis combinedly called as the eating movement. Next, also cell plasma membrane functions as exocytosis. अभी खाना खा रहा है but release करना भी जरूरी है ना students. Without just dumping the food inside the cell, is it okay? No, the release of food is also necessary, so that process is called as exocytosis. Okay, what is that process called? Exocytosis. That means cell is vomiting, removal of toxic substances. Exocytosis. In this case, what is the cell doing? Removal of toxic substances from the cell. That means cell उल्टी कर रहा है. इस केस में what is cell doing? He is vomiting. Is the functions clear of the cell membrane? See, first they will help in the transport of the molecules. Next, compartmentalization. Then the intercellular junctions present. Interaction with the other cell. The endocytosis, eating movement of the cell. In that eating movement we saw two, that is phagocytosis and pinocytosis. Phagocytosis also called as cell eating, that cell eating, and pinocytosis cell drinking. Yes, so apart from cell eating and cell drinking, release of toxic substances are also necessary. Removal or release of toxic substances, so that is nothing but the exocytosis. Okay, next is cell membrane types of transport. As we see that the function of the cell membrane is the transport of molecule. Yes, so there are two types of transport involved here: passive transport and active transport. Yes, so passive transport में क्या होता है students? It is passive में there is no ATP breakdown. Please write it down. In types of transport, in the passive transport, ATP breakdown is seen. Okay, what happened here? The molecules flow from the higher concentration to the lower concentration. Okay, the neutral solutes will enter through the simple diffusion. There are two types. It is two types. Passive transport कहाँ दिखता है? In diffusion and the osmosis. सिंपल डिफ्यूजन में क्या होता है स्टूडेंट्स? मूवमेंट ऑफ द न्यूट्रल सॉल्यूट्स अक्रॉस द मेम्ब्रेन, एंड ऑस्मोसिस मूवमेंट ऑफ वाटर बाय डिफ्यूजन अक्रॉस द मेम्ब्रेन. Yes, please remember कि passive यानी nothing but the diffusion process. What is happening? There is no energy required, no ATP breakdown is done, and the solutes or the molecules across the membrane along the concentration gradient move from the higher concentration region to the lower concentration region. Is it clear? Now in case of active transport, that is ATP breakdown is done here. Yes, what is happening? Energy is required. ATP breakdown. ATP breakdown. एक्टिव ट्रांसपोर्ट में हमें एनर्जी लगती है स्टूडेंट्स, वी रिक्वायर्ड एनर्जी, whereas in passive transport we doesn't require energy. The molecules or the fluid flows from the high concentration to the low concentration. Active transport, as ATP breakdown is to high concentration, it is also called as the uphill movement. Yes, example is sodium potassium pump. Yes, see, movement of molecules across the membrane against the concentration gradient, that is from the lower concentration to the higher concentration with the expenditure of energy, that means ATP is utilized. And it is example क्या दिया है? Sodium potassium pump. Okay, right, it is the uphill movement. Yes, uphill from low to high, so the uphill movement. And passive is downhill movement. Downhill movement. Yes, ऊपर से नीचे, तो high concentration to low concentration. Yes, is it clear about everything? Clear about the plasma membrane? Now let's see about cell wall. How is cell wall? It is a non-living rigid structure. Yes, found in plant cell, and it is made up of cellulose. Plant cell में now let's see. Plant cell में किससे बनी है cell wall? Cellulose, hemicellulose, pectins and proteins. Right? Algae में किससे बनी है स्टूडेंट्स? इट इज मेड अप ऑफ सेल्यूलोज, गैलेक्टोज, मैनोज. बैक्टीरिया में किससे बनी है? पेप्टिडोग्लाइकिन. एंड फंगाई में बनी है काइटिन एंड द फंगल सेल्यूलोज से. First of all, it is what is the characteristic of cell wall? It is a non-living rigid structure found outer to the plasma membrane of fungi and plants. Animal cell में cell wall है क्या students? Is animal cell में cell wall present? No, cell wall is absent in animal cell. It is just present in the plant cell. Structure wise, it is a non-living rigid structure. Okay, in algae it is made up of cellulose, galactans, that is galactose, mannose and minerals like calcium carbonate. Okay, next, इन प्लांट्स किसमें, प्लांट से किससे बनी है? सेल्यूलोज, हेमीसेल्यूलोज, पेक्टिन्स एंड प्रोटीन्स. नाउ कम्स बैक्टीरिया. बैक्टीरिया में किसकी बनी है सेल वॉल? प्लीज राइट इट डाउन, इट विल कम एज ए एमसीक्यू क्वेश्चन. बैक्टीरिया में सेल वॉल बनी है पेप्टिडोग्लाइकिन से. Peptidoglycan. Yes, and in fungi it is made up of chitin and fungal cellulose. Okay, now how is this cell wall? First of all, it is the outermost layer. It gives protective covering. Yes, it is rigid. क्या है characteristic? Rigid. It also provides shape and structure to the cell. Yes, dead layer is present. Please write it down, students. Write down the characteristic. It's outermost, rigid, provide shape and structure, okay. Now this cell wall has cell wall of a young plant has primary cell wall that is capable of growth, okay. धीरे धीरे क्या होता है? Now let's see in a simple diagram. I will draw here or here. Okay, ये क्या है? This is the plasma membrane. PM बोलेंगे हम इसे, okay. Next, in this I will use another color, okay. Again I will use another color. Okay, and the circular region is nothing but the plasma membrane. So I have just drawn a rough diagram, students, okay. So जो रेड कलर का है that is called as 1 degree, that is the primary cell wall. What is that? Primary cell wall, okay. कैसे बना ये प्राइमरी सेल वॉल स्टूडेंट्स? इट इज मेड अप ऑफ सेल्यूलोज एंड हेमीसेल्यूलोज. Primary cell wall is made up of cellulose and hemicellulose. Next, how what is the characteristic of this cell wall? It is elastic and completely permeable. यानी किसी को भी अंदर आ दे सकता है. It allows everyone to get inside. So this was the characteristic of the primary cell wall. Now you can see green color border line. Yes, this green color line border line is nothing but the second degree wall, that is secondary cell wall, okay. Now how is this cell wall, students? It is impermeable. सबको आने देगा? No, it is impermeable. How it is? Rigid and made up of pectin lignin, okay. And the inside blue color is the third degree cell wall, tertiary cell wall. थर्ड डिग्री यानी टर्शरी सेल वॉल. अभी ये दो प्लांट सेल के बीच में यू कैन सी दिस. यस, दिस स्पेस इज कॉल्ड एज द मिडल लैमेला. क्या बोला ये स्पेस को स्टूडेंट्स? इट इज कॉल्ड एज द middle lamella, okay. This middle lamella is cemented by, it is called as cementing layer. It is called as cementing layer and it is made up of calcium magnesium pectate और and it is permeable. कैसी है वो cell wall? middle layer, how it is, middle lamella is permeable, means it allows. Now we will read this. Cell wall of a young plant primary wall is capable of growth. It gradually diminish as the cell matures and the secondary wall is formed on the inner side. जैसे जैसे ये mature cell wall, cell mature होता है, the primary cell wall what happened, it diminished and the secondary cell wall is formed towards the inside. How is this secondary cell wall? Mature cell wall है. It's a mature cell walls. Okay. The middle lamella is a layer mainly of calcium pectate, calcium और magnesium. Also calcium के साथ calcium या calcium pectate की middle lamella होता है. क्या काम है मिडल लैमेला का स्टूडेंट? इट ग्लूज द डिफरेंट नेबरिंग सेल्स टुगेदर. सी यू कैन सी इन दिस डायग्राम, ये दो सेल को क्या किया उसने, चिपकाया है. इट इज एक्टिंग एज अ ग्लू. The cell wall and the middle lamella may be transversed by plasmodesmata which connect the cytoplasm of the neighboring cells. Yes, means kya students, what is happening? Plasmodesmata, what is the condition of plasmodesmata? See, again I will draw a diagram here. Suppose this is the cell. Okay, now this, the secondary part is been connecting. Yes, yes or no? The ER of this cell is connected to the ER of this cell. I have just drawn a rough diagram. Okay, and the middle connection is called as desmotubule. Desmotubule. Okay, now in this is the condition, the cell wall, you can see the cell wall and the middle lamella, this was the middle lamella, have been transferred by plasmodesmata. Plasmodesmata is a condition which connect the cytoplasm of the two neighboring cells. It is living connection. Okay, it is living connection. Is it clear students what is the plasmodesmata condition? Next is the functions of the cell wall. What is the function? It gives shape to the cell. It protects the cell from mechanical damage and infection. It helps in cell to cell interaction. It acts as a barrier to the undesirable macromolecules. Yes, so these are the functions of the cell wall. Shape देती है. Yes, it gives shape. Next, it protects the cell from mechanical damage. It helps in cell to cell interaction. It acts as a barrier to the undesirable molecules. So that's, here we complete the cell wall. So let's quickly revise in the session what we saw today. First of all, we saw about what is eukaryotic cell. Eukaryotic cells are the true cells which have true nucleus in them. Next, they include protists. How are they? They have well organized membrane bound nucleus and organelles. Yes, they show compartmentalization. They have complex locomotory and cytoskeletal structures, and the genetic material is organized in the form of chromosomes. Then we saw the difference between animal cell and plant cell. Cell wall absent, cell wall present, plastid absent, plastid present, vacuole is small or absent in case of animal cell, vacuole is central and big. Reserve food in the animal cell is in the form of glycogen, whereas in plant it is in centriole is present and centriole is absent. Next we saw the cell membrane, cell organelles in the eukaryotic cell, which are a cell membrane, cell wall, endomembrane system, mitochondria, plastid, ribosome, cytoskeleton, cilia and flagella, centrosome and centrioles, nucleus and microbodies, out of which in today's session we just studied about cell membrane and cell wall. Yes. Next, how is the characteristic of cell membrane? It is universal, living, fluidity, asymmetric, and have patchy appearance. These lipids are mainly made up of phosphoglycerides. Yes. Then in case of human RBC membrane, we saw that this RBC membrane only has 52% of protein and 40% of lipid, and the rest is all cholesterol and oligosaccharides. Based on this extraction membrane, proteins are of two types, that is intrinsic, integral protein and peripheral protein. Integral protein are the proteins which are totally or partially buried in the membrane, whereas peripheral proteins lie on the surface of the membrane. If we see one phospholipid, it has a head is called as polar and the tail is non-polar. Polar, it is hydrophilic, that is water loving, whereas non-polar is hydrophobic, so it is apathetic in nature. Yes. Next, cholesterol is present here for maintaining the stability. This sugar chain, that is glycoprotein or oligosaccharide, is present for functioning as the antigen. Next, this model, fluid mosaic model, was composed by Singer and Nicholson in the year 1972. There Nicholson proposed a line that proteins are like iceberg in the sea of lipid. What they say, Singer and Nicholson ने क्या compose किया, that this cell membrane is made up of lipid bilayer plus protein, and the ability to move within the membrane is measured as fluidity. Okay. Next we saw what are the functions of the cell membrane. It is helping in transport, then cell growth, formation of intercellular junctions, secretion, endocytosis, and cell division. In endocytosis we saw the two, that is phago and pino. Phago is called cell eating and pino is called cell drinking movement. And apart from this, cell is removing all the toxic material present, that it is called as exocytosis. Yes. Next we studied about the two types of transport, that is passive transport and active transport. When passive transport, there is no ATP breakdown, it is from higher concentration to lower concentration, and it has two types, that is simple diffusion and osmosis. It is a down the hill movement. Whereas in active transport, we saw the ATP breakdown, that energy is required in this case. It is from low concentration to high concentration, and it is also called as uphill movement. Example, we saw that sodium or potassium pump. Now, then we saw about cell wall. We studied it as the outermost outermost and the rigid layer. It provides shape and structure. In case of plants, it is made up of cellulose, hemicellulose, pectins, and proteins. Whereas algae, it is made up of cellulose, galactose, mannose, and minerals like calcium carbonate. In case of bacteria, it is made up of bacterioglycan, and in case of fungi, it is made up of chitin or fungal cellulose. Then we saw the cell wall, the three layers of cell wall: primary, secondary, and tertiary. The primary layer is the outermost layer. It is the young plant cell, and when the cell reaches the maturity, the primary layer diminishes and secondary wall is formed. That wall is impermeable, rigid, and made up of pectin and lignin. Now these two cells have a space in between, that is called the middle lamella, which acts as a glue for the neighboring cells together. And this middle lamella, it is made up of cementing layer composed of calcium or magnesium packet, and it is permeable in nature. It helps which connect the cytoplasm of the neighboring cells. The functions of the cell that it gives shape to the cell, it protects the cell from the mechanical damage and infection, it helps in cell to cell interaction, it acts as a barrier to the undesirable macromolecules. So that's all for today. We studied cell membrane and cell wall. Thank you.